Hello everyone. This is Kendra Vidyalaya Sangathan JIT Bhubaneswar's initiative. We are presenting digital videos for chapters in class 11th and 12th. Hello students. Welcome to the next part of our ongoing series on biotechnology, principles and processes. As you know, the chapter has been divided into 5 learning units, and we are on to the second unit where we'll have discussions about tools of biotechnology, and specifically that will include restriction enzymes and gel electrophoresis. As we know, the units given in this chapter: biotechnology, basic principles and history and its overview; tools of biotechnology, restriction enzymes and gel electrophoresis. Tools continues in the form of cloning vector and technique for making host competent. Then we move on to processes of recombinant DNA technology, including isolation of DNA, cutting of DNA, and gene amplification using PCR. Then process of recombinant DNA technology. Next part, we'll discuss about insertion of recombinant DNA into host and obtaining foreign gene product. So in our journey to master the concept, we are on the second rung of the ladder: tools of biotechnology, restriction enzymes, and gel electrophoresis. Before we start, let us share the expectations, what you should know already. You should be knowing the basic structure of DNA as double helical structure. You should be also aware about the replication mechanism of DNA. You should know the chemical nature of bonding in DNA. You should also know the nature of charge on DNA and the concept of potential difference, that is cathode and anode. After watching this video lecture carefully, you'll be able to explain the role of endonuclease and exonuclease enzyme, describe the nomenclature of restriction endonuclease, explain working of restriction enzymes in biotechnology, enumerate steps in creating recombinant DNA molecule, and explanation of gel electrophoresis construction and working. Let us first of all focus on the tools of biotechnology. There are many tools, out of which we classify them one by one. There are enzymes available, and enzymes can be cleaving enzyme, which includes exonuclease, normal endonuclease, and restriction endonuclease. Lysing enzyme, the one which breaks down the cell wall, lysozyme, cellulase, and chitinase in different types of cells. Synthesizing enzymes, including reverse transcriptase, DNA polymerase, and joining enzyme or DNA ligases. We'll be focusing though on restriction endonuclease and DNA ligases. Then we talk about gel electrophoresis, the technique which is used to separate the DNA fragments. Then cloning vectors or vehicular DNA, which includes plasmids, cosmid, bacteriophage, and some others. And then competent host, technology to make competent host. And this includes some ways like chemical method, electroporation, gene gun, microinjection. We'll be reading in subsequent parts. First of all, focus on enzymes in biotechnology. The major enzyme categories are cleaving enzyme, lysing enzyme, synthesizing enzyme, and joining enzyme. As the name suggests, cleaving enzymes cleaves or cuts the DNA. It includes exonuclease, which mainly cuts from the terminal ends of DNA. Normal endonuclease, found in normal eukaryotic cells, which cleaves the DNA from within. And restriction endonuclease, isolated from bacteria, and it has got specific sequence where it cuts. We'll see the significance. We have got lysing enzyme. In the processes, we'll be reading about the role of these enzymes: lysozyme, that degrades the cell wall of bacteria; cellulase, digesting cell wall of plant cell; and chitinase, digesting cell wall of fungal cell. Synthesizing enzymes, sometime we need to engineer retroviruses So there we need reverse transcriptase, or if we have got cDNA, then we have got DNA polymerase, and the joining enzymes or the glue, the biological glue, DNA ligases. First of all, we focus on cleaving enzyme, exonuclease, normal endonuclease, and restriction endonuclease. Exonucleases are the one that cleaves DNA from the terminal ends, as already mentioned. So they'll be cutting the DNA from the ends. They have got capability to cut the phosphodiester bond, but only from the end, either carboxy end or amino end. The normal endonucleases, these are the one that cleaves the DNA from within by cutting the phosphodiester bond. So they are also found in eukaryotic cell. We talk specifically about the third and important enzyme, which is used, or rather say, because of which this biotechnology or recombinant DNA technology became possible. Restriction enzymes are endonucleases obtained from eubacteria and archaea that recognizes a specific DNA sequence. The nucleotide sequence recognized for cleavage by restriction enzymes are called as restriction sites. We'll be reading in details. Let us see how exonucleases act. If you look at the sequence, you will find, see, these are exonucleases that cleaves the DNA from the side. Let us see how normal endonuclease acts. Normal endonucleases cleaves the DNA from within the DNA. And you can see it is being cleaved, right? So why endonuclease is needed? That's another point of discussion. So if you look at their working mechanism, we can understand by this illustration that exonuclease activity yields excises at terminal end, whereas endonuclease activity produces excision between terminal ends or in between, right? Now we have a look at restriction endonuclease. These enzymes are isolated from eubacteria and archaea, and they recognizes a specific DNA sequence. And these sequences are palindromic. Palindrome, which reads same from both front and end, as for example, Malayalam, M-A-L-A-Y-A-M, Malayalam. As far as discovery is concerned, the restriction enzymes were first discovered by Arbour and Smith and Nathans. The first enzyme to be discovered was HIND2 in Bacterium Haemophilus influenza. Actually, Arbour find out how E. coli protects itself from the action of bacteriophage. What happens? These bacteria does not have a defense mechanism and they are attacked by bacteriophage virus. So what happens? They have got a restriction mechanism. You'll find there are some enzymes found inside, set of enzymes, two enzymes, one is methylase, another is restriction endonuclease. Methylase masks the recognition site by adding a methyl group so that the restriction endonuclease does not recognize its own DNA and should not degrade it. And restriction endonuclease available for anything that comes from outside or foreign genome. Let's have a look in the animation. As the bacteriophage comes, attaches itself to the bacteria, injects its genetic material, which copies. So now comes the role of restriction enzymes. Restriction enzyme will come and degrade these DNA. Why? Because the recognition sites are not masked. So this is how it restricts the growth of bacteriophage, or rather say action of genetic material of bacteriophage. Hence it is called as restriction endonuclease. You can see in the legend, bacteriophage, viral genome, bacterial genome with methylated base protection, the enzyme methylase, and restriction endonuclease. You'd have seen the names of these enzymes. They are very funny to look at. EcoR1, HIN2. So funny. But they are not funny. They follow a very particular convention. Just for example, we take EcoR1. In EcoR1, the E is written in capital. It is the first alphabet of the name of the genus Escherichia. CO, the next two letters from the species name of the bacteria coli. Then comes the first letter from the strain. The strain we talk about is RY13. And the last Roman 1 indicates the order in which the enzymes were isolated from that strain of bacteria. That's how we get the name EcoR1. Right? There are some other examples. Let us take a small test. The genus is given, species is given, the strain is mentioned, the order of discovery is mentioned. Now you need to take pen and paper and write down the name of restriction enzyme in order to find out whether you have understood the concept or not. I'll read it out for you. Escherichia coli, strain RY13, first enzyme to be discovered. Next example is Bacillus amyloliquefaciens, strain is H, order of discovery first enzyme. Haemophilus influenza, strain is D, order of discovery third enzyme. Staphylococcus aureus, strain is 3A, and order of discovery is first enzyme. Let us see if you have got it correct. Take your time. Simple convention. Remember the last slide. How did we name a restriction enzyme? The first one was already done for you. Yes, you are right. EcoR1. Now, if you have done it, let us click here to know the right answer. Wow. If you have written in the same way as the answer has appeared, then you have understood perfectly well how to name a restriction enzyme. EcoR1, BAM-H1, HIND3, SO3AI. This is an important question that is generally asked in examinations as well. Naming the restriction enzyme. Now, we look at the specific sequence or restriction site. As I have already mentioned, the restriction site is a palindromic site, specific site recognized by the restriction enzymes. And here we take example of EcoR1. The EcoR1 recognizes the site GAATTC. You can see it is mentioned in red, and you see from the both side of this DNA, it reads the same GAATTC. Right? So now, how restriction enzyme works? They can approach from this end or here and provide a kink, and then they cut the hydrogen bond in between and leaves two parts with sticky ends. Right? What I mean sticky ends. And if same type of DNA fragment is produced in two different sources of DNA, then they can be joined together. So this acts as joining point. This sticky end produces joining point. I should also mention there are certain cases where blunt end is also produced, but that is beyond the preview of our discussion at this moment. But till the point, let us keep down to the recognition site of EcoR1, which happens to be GAATTC. This is one diagram that you have already seen in your textbook. How this enzyme cuts. Enzyme cuts both DNA strands at same site. This is the vector DNA. You see GAA-TTC and the foreign DNA at the same site, right? And both sticky ends are produced on vector DNA as well as on the foreign DNA. Now it facilitates complementary bonding of these two, thereby creating a recombinant DNA molecule. This is what was most significant, creation of recombinant DNA molecule. The joining points, right? So now we talk about another enzyme, DNA ligase. It's a specific type of enzyme that facilitates joining of DNA strands by catalyzing formation of phosphodiester bonds, right? And as you can see, the sticky ends are there. When they approach, the hydrogen bonds are being formed and ligase forms the bond in between the fragment, thereby joining or sealing the fragments. Now we talk about one more technique. We have got the cleaved DNA, how to separate them, because it's very, very small to look at. We cannot just look at through our unaided eyes. We have got a technique in biotechnology which is used to separate DNA fragments on the basis of their size and charge. This is called as gel electrophoresis. You'll find here we'll talk about agarose gel electrophoresis. You find there is a tray which has got agarose gel cast. Here we have got certain wells created with the help of combs, and the two ends are connected to the power supply. One is cathode, another is anode. As we know, DNA fragments are negatively charged. So if a potential difference is applied here out, what will happen? The fragments will start moving towards anode or positive end. So two things are important here out. The smaller DNA moves to farther part. It moves farther as compared to the bigger ones, number one. And number two, the movement is always towards the anode, right? So this is another conceptual clarity you should have while studying gel electrophoresis. So Tiselius devised this in 1937, and the DNA fragments are negatively charged and they can be separated by forcing them to move towards the anode under electric field through a medium or matrix. The commonly used matrix is agarose, which is extracted from seaweeds. It's the natural linear polymer of D-galactose and 3,6-anhydro-L-galactose. The DNA fragments separated out according to their size through sieving effect provided by the agarose gel. So smaller fragment will move farther. Separated DNA can be visualized after staining the DNA with ethidium bromide. And when the DNA is stained with ethidium bromide, this stain binds with the bases and produces an orange illumination. They produce orange bands if they are washed under trans-illuminator.
Here are some further readings.
Under
you can
a UV chamber.
And also animations.
Now the separated bands of DNA are cut out
you must see
and extracted from the gel
revolution.
piece. This step is called as
it's a TED
elution.
Talk video which talks
And
about
purified DNA
the
fragments
invisible
are
revolution
used to
as
form
created by biotechnology.
recombinant DNA, which are joined with the cloning vectors. So today we have seen about two important tools of biotechnology: the enzymes, restriction endonuclease, ligase, and a technique to separate the fragmented DNA, which is nothing but gel electrophoresis. So biotechnology uses many tools to produce recombinant DNA. Restriction enzymes are one such tool that cleaves DNA at particular site, such as to allow creation of recombinant DNA molecule. Arbour and Lind discovered restriction enzyme.
Thank you for watching it so patiently.
Hint2 was the first enzyme to be discovered and isolated. Cohen and Boyer created first recombinant DNA by using antibiotic resistance gene from Salmonella typhimurium and inserted into E. coli. And restriction enzymes follow a definite naming convention. The first letter is written in capital and derived from the genus name. Next two letters in small from specific name. Next letter for strain and Roman numbers to show order of their discovery. As for example, Eco R1, Escherichia coli RY13, and first enzyme to be discovered. Now, the cleaved DNA are to be separated. That is done using gel electrophoresis. It's a technique for separating DNA fragments based on their size and charge. And the sample DNA is cut into fragments. So now what happens? The DNA fragments being negatively charged when placed under a potential difference in the agarose matrix, it moves towards anode under electric field. And agarose is the common matrix used. So that's why it is called as age or agarose gel electrophoresis. The DNA fragments separate out according to their size because of their sieving property of agarose gel. Hence, we have seen the smaller the fragment, the farther it will move. Now let us get some assessment, what we have read and how far we have understood. I'll read the question for you. Be ready. Take the pen and paper and start writing the answer. The DNA fragments produced by the use of restriction endonuclease can be separated by polymerase chain reaction, gel electrophoresis, density gradient centrifugation, any of the above. Move to the next question. Which of the given statement is correct in the context of observing DNA separated by agarose gel electrophoresis? DNA can be seen in visible light. DNA can be seen without staining in visible light. Ethidium bromide stained DNA can be stained in visible light. And ethidium bromide stained DNA can be seen under exposure to UV or ultraviolet light. We move to the third question. Restriction in restriction enzyme refers to cleaving of phosphodiester bond in DNA by an enzyme, cutting DNA at specific position only, prevention of multiplication of bacteriophage in bacteria, all the above. Next question talks about the role of DNA ligase in construction of recombinant DNA molecule is formation of phosphodiester bond between DNA fragments, formation of hydrogen bond between sticky ends of DNA fragments, ligation of all purine and pyrimidine bases, none of the above. Now we have gotten small exercise. Match the items in column 1 with those of column 2. Let us see. Seaweeds, staining of DNA, separation of DNA fragment, elution. And we have got in column 2 gel electrophoresis, source of agarose, isolation of DNA from the gel, and ethidium bromide. If you have done it, let us go back and check the correct answers. If all the answers are correct, you have learned the concept beautifully. If you lack somewhere, no worries, go back, watch the video, read your book, and revisit the question. Let us find out the correct answers. Yes, separation of DNA is done by gel electrophoresis. After staining the DNA with ethidium bromide, it can be only seen under exposure to UV light. The prevention of multiplication of bacteriophage is done through restriction enzyme. That's why it's called as restriction endonuclease. And ligase forms phosphodiester bond between two fragments. Seaweeds are isolated source of agarose. Staining of DNA done by ethidium bromide. Separation of DNA fragments through gel electrophoresis. And elution means isolation of DNA from the gel. I hope you would have checked your progress and you would have done well, reasonably well. Take some home assignment. The questions already displayed. You need to write the answers neatly in your notebook and submit for correction to your respective mentors. If you have not done, I am repeating it again. Please take it down. You can revisit the concept. There's a wonderful book, Molecular Biology and Biotechnology, edited by John M. Walker. There's some website links. You can also view the animation of gel electrophoresis and animation on restriction enzymes. So the next topic. We have seen biotechnology, basic principles and history and overview. We have read about tools of biotechnology, restriction enzyme and gel electrophoresis. In the coming video, we'll be reading about tools of biotechnology, cloning vectors, and making host competent. So keep tuned and get more information on these topics. This video lecture was presented by Mr. Rajiv Ranjan Singh, PGT Biology, Kendra Vidyalaya, Air Force Station, Kumbhagram, Silchar region. The task force behind this